Okay, so we know from work in my lab, from Veronica Van Hennigan's lab, and a couple other labs around the world, that about 2/3 of all aniridic patients are going to have a mutation in the PAX6 coding region. So remember, the PAX6 coding region is only the part of the gene that makes the protein. And so I've changed the schematic on you this time, so you're seeing a stick diagram or a structure diagram shown here, in which the start of the protein is to your left side of your screen, and the end of the protein is at the right-hand side of your screen. The paired domain and homodomain represent the DNA binding domains that allow PAX6 to work. The green region is called the PST region. It's the part of the protein that I showed you earlier that allows other proteins to work with PAX6 to control eye development. This slide is somewhat old now. At the time I made it, there were 286 different types of mutations that we knew about. This number is now closer to 300. But the essential information in the slide is correct. So for somebody that has classic aniridia, or a typical presentation of aniridia, we are 66% certain that they're going to have a mutation that affects somewhere in the protein. So now we need to start talking about what types of mutations one might see. And so this is just a slide defining some of the types of mutations that we have to think about. Nonsense mutations are simply single base pair or single nucleotide changes. So one letter has been changed, such that it now creates a stop codon in the open reading frame. And so I'll show that to you in just a second. The next common one is a splicing mutation. So remember that the genes are made up of introns and exons. A splice mutation affects the way that information is put together. A frame shifting, insertion, or deletion is simply going to change the reading frame by either adding or subtracting bases. Now, the reason that it's not that we call these insertions or deletions as frame shifting is because remember the amino acid code is in triplet. So if it's not divisible by 3, it's definitely going to have an impact on the open reading frame. If the insertion or deletion is divisible by 3, then it will probably shift the reading frame in some way, shape, or form. And we're going to see all of these in examples in just a couple slides. Last but not least, there are missense mutations in which a single base has changed such that you change one amino acid in the protein to a different amino acid. So it doesn't prevent the protein from being made, but it can change the way the protein can function. And last, run-on mutations. In this case, a stop codon has been converted to something else, and we're going to see examples of all of these. So this is taken from a review from a couple years ago. The majority of PAX6 mutations, but the information is still current. The majority of PAX6 mutations associated with aniridia do not make functional PAX6 protein, and so we're going to be talking about primarily about nonsense mutations, splicing mutations, and frame shift mutations. So in a nonsense mutation or a frame shift, essentially there's a change in the DNA, shown here by the stop sign, that is then, because the RNA is a faithful copy of the DNA, is put into the mRNA, so the message, but the message, because it has an inappropriate stop, can no longer be made into protein. And so a nonsense mutation is a change in the DNA that prevents PAX6 from ever being made. And if PAX6 is not made, it's essentially as if it was never there. If we were to actually look at the sequence, so in this case, I have two, and so we're going to see several slides like this. I have the normal PAX6 sequence at the top, and I have a mutation shown on the bottom. We're going to pick on exon 5. So as we work from left to right, you'll see exon equals number 4, exon equals number 5, exon equals number 6. So I'm showing you the exons that come immediately before and immediately after exon 6. There's after 5. There's 6, and this is the region that we're interested in, this GAG that's in blue. if there is a single nucleotide change shown by the red T at the bottom of your screen, then what should have encoded the GAG, which should have encoded for E, is now converted to a stop codon, which is denoted by the asterisk. And so this protein, if it were being translated, would start with the M and end with the V in a place that it should never end. And so this protein essentially is not going to work. The other way this could happen is through a frame-shifting mutation. So in this case, we are playing with the same GAG shown up here at the top. In this particular mutation, the area denoted by the pink brackets was deleted. So the nucleotide, that single G that's up here, was deleted from the DNA. As translation begins with this methionine, the ATG, it's going to read in triplet all the way through, but when it gets to this GTA shown here, which normally encodes for V, valine, the next triplet has to be AGC instead of AGAG. So the cell happily reads AGC. It makes the amino acid serine, S, instead of the amino acid leucine, L. And the next triplet is the stop codon TAG, and so it stops. Again, a functional PAX6 protein cannot be made. Now, let's move on to an in-frame mutation. These very often will change the PAX6 protein, but they do not necessarily destroy the function. And so there are very few of these that are involved in aniridia. But they do occur. And so, again, we're playing with the same presentation. So we have our GAG shown up here at the top, which normally makes for E. And in our mutation in exon 5, shown down here with the bracket, that triplet, that GAG, was physically deleted, and so the cell reads GTA and then jumps to CTA. If you compare that to the normal sequence, we still have V and L, but we're missing the E that normally comes in between. So that could have an effect on protein function. Now, one of the most common types of mutations that we've been finding are the splicing mutations. The second most common, to the nonsense mutation. So a splicing mutation in this case affects the way the gene is put together. And so now we have to back up a little bit. Genes like PAX6 are divided into exons. And in this example, I have a gene that has 3 exons. In between the exons are DNA sequences that are called introns. Intronic sequence is simply the DNA between 2 exons. At the left-hand-most side, at the top of your screen, there's a little 5', and then I have a little pink bubbles there that I just marked. That denotes the region of the gene that controls whether the gene is on or off. These are called regulatory sequences. They're often found 5' or upstream of the gene. They can also be found, so 5', they can also be found internal to the gene or 3' to the gene. The important thing about these structures is that they all control whether the gene is turned on or off. Okay, now we need to go back and pay attention to our exonic sequence and our intronic sequence. PAX6 is a very complicated gene, even by most gene standards. It is large. It actually encompasses about 25, you could consider 25 miles of highway. In our language, it's 25 kilobases. It has 3 promoters. We call them P0, P1, and P alpha. P alpha is an internal promoter that has a very special set of conditions that we'll come back to. We know that it has 16 exons, and so there are 13 exons. You can see them 1 through 13, plus an exon 0 and an exon 5A. What you don't see here is the alpha exon, which goes with that promoter. So PAX6, the important things here is that PAX6 is a complicated gene. It has 3 promoters, P0, P1, and P alpha. The 2 upstream promoters, P0 and P1, are the 2 that are going to control most of gene expression. We'll see that in a moment. The exons, as you can see, are distributed over the entire region, although some introns can be very small, such as that between exons 3 and 4, or the exons can be fairly large, as between exons 9 and 10. PAX6 sits on chromosome 11p13. It's flanked by the Wilms tumor gene on one side and FSHB, or follicle-stimulating hormone beta, on the right side. There are actually other genes that are situated in here, and so this tends to be a fairly gene-rich region. The only reason that is important is if there's a deletion that takes out Wilms tumor to somewhere over here. Not only do you have aniridia from PAX6, but you also have other problems associated with the Wilms tumor gene and some of the other genes that are situated in this region. This is the Wagner syndrome, and it's a Wagner deletion. Now, I said that PAX6 was complicated. So, transcripts for the PAX6 gene actually encode for 3 types of protein. We're only going to pay attention to 2 of them today. Transcripts that start from either the P0 promoter, shown here, or the P1 promoter, shown here, encode for the protein that we've been talking about, and that's the PAX6 protein that has 2 DNA binding domains, the paired domain and the homeodomain. What you see in B, both there and also for P1, are the transcript maps. And so the reason that there are broken, or the arched bars between the exons, so there's the arched bar between our exons, denotes the fact that when this is being transcribed, information from exon, in this case 0, is spliced to exon 2, is combined with 3, 4, 5, and so forth. That's how we get the message that encodes for the full-length PAX6. Transcripts that begin with the internal promoter, like P0 over here on the left, also splice, but you can see in this case we're going to include exon 1 to 2 to 3 to 4. So the important thing is that when a cell is making a transcript, it has to do 3 things. It has to recognize the promoter and turn the gene on. It has to read through and faithfully replicate all of the DNA or all of the information contained between the promoter and the end of the gene. And then it has to assemble all of the exons together in the appropriate way to make the appropriate message. And so the other thing that you'll note is that there are different codings. And so exons that are open make for non-coding sequence, and that's the sequence I showed you earlier. We looked at the PAX6 transcript. So this is the untranslated region, whereas exons that are shaded are the ones that actually make for protein. Now, while we're on this slide, the other thing that I need to point out is this internal promoter. We're not going to talk about it very much today, except that it exists, and it makes a different type of PAX6 protein. In this case, it's missing the paired domain entirely. We don't know very much about it, except that we know that it's important for eye development. Now, how does a splice mutation affect how the protein is actually made? So in this case, again, we're looking at the normal splicing pattern of PAX6. So in this case, I'm going to use the first promoter, P1, splices to exon 2, to 3, to 4, 5, and so forth. This is going to make a protein that has the paired domain, the homeodomain, and the transactivation domain normally. If there's a mutation